Hi everyone, how are you doing? So we're going to be talking about our next topic, which is enzymes. So when you are looking at the PowerPoint, I want you to make sure that you are using the Microsoft Word document to define the words that are listed here. And throughout the PowerPoint, you should have all the definitions that are listed. But if you happen to use Google, make sure you're using enzymes as a reference when you're doing a Google search on any of these vocabulary words. If you look up substrate, it might come up with something that's related to, rocks or something like that, and not enzymes. So make sure you're doing Google search and using enzymes as your goal in doing the vocabulary definition. Okay, let's get started. So enzymes are proteins, and they act as biological catalysts and are essential to the functioning of a cell. So what is a catalyst? A catalyst is something that speeds up reactions. So if you think about it, if I tell you you need to go to California in 2 days, there may be many ways that you can get there. Maybe you walk, maybe you drive, maybe you take a plane. But remember, I said 2 days. So realistically, how are you going to get there? You could technically drive in 2 days, yes, but definitely not walk. So flying is going to be the best option for you to make it in that time. That's what catalysts are, is they take you to the place you need to go in a speedy time frame. So it might take you 5 hours to fly and maybe, 48 hours to drive, and I don't know how many days to walk, but I'm quite sure it'd be very long. So enzymes are needed for catalyzing and speeding up those chemical reactions. These chemical reactions could happen on their own, but they might say, let's take, in the lifespan of Earth's history, 10,000 years or 50,000 years. So they're going to take a very, very long time. So humans don't have a long time to synthesize body parts. They don't have 10,000 years. So they need to get to that destination in a much quicker way. And that's how living organisms work. So catalysts are essential to allowing those organisms to survive. So there's a definition. And catalyst is going to increase the rate of a chemical reaction. The chemical reaction could occur, right? It could take, maybe 5,000, 6,000 years for something to break down or to be made. But humans or any living thing doesn't have that time span. So catalysts are essential for living things in order for them to have those same or similar reactions happen. Most enzymes are proteins. There are some exceptions, and we'll talk about them when we do DNA synthesis. Ribozyme is an example of an RNA enzyme, if you want to call it that. It's essential in joining amino acids when we're making proteins. So we do have some exceptions to the rule, but on average, we can say most enzymes are going to be proteins. Enzymes are going to do two things for us, right, in general. They're either going to digest, which we call anabolism, or if they synthesize or make compounds, that's called catabolism. But remember, enzymes are only going to do one type of function. Even though I wrote on the board that they can do, we can have that reaction happen forwards and backwards, digestion and synthesis. That is true, but it's only done with one type of enzyme, right? So one enzyme can only do one type of reaction. This is the induced fit model. The induced fit model is showing how enzymes are able to function and do their job. Remember, their job is either to digest and break apart or to put together. So here's some vocabulary words you still need to define. When you look at the enzyme is represented green here, and then there's a shape of the enzyme, yes, on the outside, but more importantly on the inside. That is called the active site. The active site is where the substrate, which is what the enzyme is working on, we call that the substrate, is going to go and fit into the enzyme. So there's all these different areas on the enzyme, but the substrate is going to just fit into the active site. And you can kind of see it almost fits in, right? The shape is going to fit in there, just like a key would fit into a lock. So then the enzyme is going to create something called the enzyme substrate complex. And there's a lot of reactions going on there in the sense of like bonding and attraction. And so it's not just, like a puzzle piece fitting in. There's a chemical reaction going on here in bonding. And the enzyme is going to do its job. In this case, it's doing the job of digestion. And we know that because we see two pieces broken apart here. The end part we call is the product, right? We went over that in class the other day. So substrate is the part that you start with, which is the reactants. And then the arrows, remember, you're making or yielding something. And here you're yielding or making end products through the process of digestion. And if you note that this shape right here looks like it may be even further digested, but this enzyme is not going to do that job. It's only going to do the job for this particular substrate. So when we look at large macromolecules, they actually are digested by multi-enzymes, and it's not just one enzyme that does the job. Enzymes are very, very specific, and they're specific because the active site will only fit with one substrate. All right, so the function of the enzyme, here's a 3D model, a molecular model of a enzyme called catalase. And here we're just going to draw it simple. That's why we have these shapes right here. And you'll see enzymes more of like a blob than like this. And the active site right here shows you that it's actually more of a bonding aspect than it is like a puzzle piece fitting in. But to make our point, we use that puzzle piece to fit in to show clearly that the enzyme is using the active site in order to bond, or bind, I should say, to the substrate. So the shape is really, really important, and the shape is the point in why we draw the enzyme the way that we do. And don't forget that proteins are made up of what? Monomer. You should know this now. Good, if you said amino acids. So all these amino acids are going to bond and bind together. They're not going to be a long, straight chain. So we talked a little bit about macromolecules not just being like a long backbone, even though fats are. Proteins tend to have a three-dimensional shape. And those three-dimensional shape is going to determine what the active site looks like, and then, of course, what that substrate is that it's going to be working on. So we say that enzymes are very specific, one enzyme, one function, and that's because the active site is very, very specific. Remember, it could actually just be the difference between, hydrogen or an oxygen. Remember, we talked about that certain starches can be digested by humans, such as starches that we find in potatoes or in corn, but in plants, the cellulose, that starch is not, and it's only literally one oxygen difference that changes that. And the enzyme recognizes that, just that one oxygen, and will not digest it. So here we're going to go back to our analogy with going to California. These are chemical reactions that can occur without enzymes. It's called activation energy. The amount of energy needed for a chemical reaction is called activation energy, and it actually is lowered with enzymes. So when a lot of students look at this right off the bat, they say, okay, if I tell them to pick which one is the one with the enzyme, if you have two reactions, they always say, oh, this one, because they always feel like it's a catalyst, it's getting more, or it's speeding up. But it is speeding up in the time, but we were talking more specifically about the amount of energy that's needed. So if you think about it, if you had to walk to California, it's going to take you a long time, a lot of energy, a lot of calories, right, to get there. Even if you had to take, even if you drove to California in 2 days, it would still take you a long time to do. You're going to have to stop and get some food, and maybe maybe go to a hotel, and you're going to have to walk and, fill your gas tank and things like that. It's going to take you a long time. But with the plane ride, you could just, let the pilot do all the work. You're going to sit back, you're going to maybe sleep, maybe watch a movie, but you're really not going to spend any energy at all in that travel. So that's why the analogy works here, is that the amount of energy used to get to California, you don't use a lot with the plane, right? And so this is the enzyme, right? Is it lowers the amount of energy needed for the same reaction to happen, and it also happens in a quicker time span. So lower energy, quicker time, and again, in a time that is essential for living things in order for them to work properly. These are some examples. You don't have to memorize them, but they're just some key parts here that will help you if you do read anything about macromolecules. Remember, we said anything with an OSE is a sugar, and anything with an INE is an amino acid, like lysine, glycine, cytosine. Sugars, glucose, fructose, sucrose, so they end in those endings. So ASE is what we use to describe an enzyme. So salivary amylase is found in the salivary glands, and amylase again is the enzyme that's going to break down starch. And then sometimes the beginning also helps you out, like peptidase, right? That breaks down enzymes that break down peptide bonds. Nuclease breaks down nucleic acids. Lipase breaks down enzymes, break down lipids, right? And then all of these enzymes, not only is their active site so essential for their function, but we're going to find out that their reaction rate is also controlled by temperature, by pH, and the amount of enzymes that you actually find in the organism. So we call that substrate concentration. So we're going to watch videos on this, so you'll see quickly in more detail than words would ever describe how enzymes are actually working. Because again, it's like a machine working. It's, you could look at a hammer and say it's a hammer and define what it does, which is what we're doing now with the definition of the PowerPoints. But in order to see how a hammer works, you need a video or maybe someone to demo how a hammer would work. It's just a little bit easier to see and understand than just to write the definitions. But I do want you to be comfortable with any of these definitions, because this is really what confuses a lot of students when they talk about macromolecules, is it tends to be very heady and wordy, lots of definitions for you. So enzymes are going to work just like we do, right? We have optimal, right, conditions where we work the best, right? Maybe it's we have the right snack in front of us, we had the right amount of sleep, right? We need things in order to work at our best. So enzymes are the same thing. They're machines. They're working for us. They have to be the right temp. So it's no surprise that we're the right temp that our enzymes work the best at, 98.6. And the pH may vary throughout your body to have enzymes work functionally. And we find that sometimes when people get sick, and if their pH is a little bit off, that could even make them more sick because their enzymes can't function and do their job. So that's always something we're concerned about, especially the blood. The blood should be a more of a neutral pH. So if it becomes acidic, we worry that they might pass away from that, the acidosis inside their blood. So we're always concerned about our enzymes and our function, right? And that sometimes will determine if we have disease or illness. Drastic changes that occur when enzymes are the wrong shape, we call that denature. So literally it like melts away. So just imagine that hammer that you're physically using to make something like a building. The hammer, let's say you throw it in, I don't know, 3,000 degrees. I don't know what it takes to melt metal, but 3,000 degrees and it melts the metal. The longer you leave it in there, the more deformed it's going to become. That's what we mean by denature. It deforms the actual three-dimensional molecular structure of the enzyme. So not only is it temperature, but pH. I'm not sure if you're familiar with this, but if you use a different pH, you can also change the effect of a particular protein. Just think of an egg if it's raw, right? An egg is a lot of protein and it has a physical shape. So if you look at a raw egg, and you heat it up and you cook it, it will change its functional shape, right? So that's why a raw egg versus cooked egg looks very, very different, because you're changing the functional proteins. You're actually denaturing them, and that's why they're easier to eat. If you do that with pH, most of us don't cook, but if you put something in an acid, right, or a base, like if you put pickles I think are in vinegar that's very acidic, it changes, again, the texture and taste of the cucumber into a pickle, right? So pH can actually also denature proteins. And that's why also foods could be preserved in different pHs. So like if you put a cucumber in vinegar, it might last, 3 years in a jar. And that's because none of the enzymes in the bacteria can actually survive that pH, and therefore they can't grow. So it becomes a sterile environment when you change the pH. And that's because bacteria cannot grow and their enzymes are not functional. So it happens more often. We just don't think of it that way, but it's kind of interesting, right? All right. And also substrate concentration, we'll talk a little bit about this, but if you have the higher the concentration, the more reaction rate you have, but then there's that like limit level, it's like just how fast could you run, you can always increase your speed with optimal, conditions, but there's going to be a max that you have. And that's just the way enzymes work as well. Okay, so it's limiting, right? So you keep on dumping it in, right? But so enzymes will only work at a certain reaction, certain amount, a certain concentration. All right, so let's look at the word optimal. So optimal tends to be a tough word for some, but it means the best. Whoops, let's go back. There we go. So optimal means the best. So if we have the optimal pH, remember pH is 0 through 14, even though it's not shown on this diagram. 7 would be right here, and 7 would be neutral. So depending on the enzyme is going to have a optimal or the best pH and temperature that it works at. So what is the optimal pH for enzyme pepsin? Remember, optimal means the best. So the optimal part would be the highest point or the peak inside the graph. So if we look at pepsin, there's our point. We follow it all the way down. We find the optimal pH or the best pH would be pH of 3. So the good thing is on the regions, this is just grabbed from a regions question. Obviously, you're going to have like a reading, about enzymes. You don't have to memorize anything about the enzyme names and what they digest or synthesize, but they do want you to be familiar with these concepts. What is the optimal pH for trypsin? So here we have the best. We followed all the way down and we see that it's 8, okay? Now here's another question. If the pH is 6.3, right, so let's mark it right there. Which enzyme would work the best? There we go. So if you said trypsin, you're right. And the reason why it's not pepsin is because pepsin stops working at the pH of just slightly under 6, or 5.8, let's say, and then it stops working. So the pH is determining its effectiveness and how well it's able to work. So you can see it still works at these different pHs, but it works the best at this particular pH. So we find that the body needs to maintain homeostasis in having the right pH in the right area. So like your stomach is extremely acidic, and that acidity is allowing certain digestion to take place, like pepsin is actually found in your stomach. And you have a couple of other enzymes that are found in the stomach that also work very well at an acidic pH. So we find that there's a reason why you have different pHs in different parts of your body, and that's, again, for your enzyme reaction rate to be the most effective. All right, so this was an exit activity last year, and I'm going to keep it here. This is going to help you for the activity we're going to have in class. So this will be on the board for you to help you with, and we're also going to have an enzyme cutout activity. Everything's going to be cut out for you. You're just going to have to assemble the pieces. So let's just go over what this would look like. This is called the induced fit model. Induced fit model is where you have the enzyme actually fitting into the substrate. The substrate here is called the enzyme substrate complex. And then at the very end, you have your enzyme digested into 2 products. So we would say that this is an example of an enzyme doing digestion. All right, so we're going to do review with our enzymes, and that will be done with the animations, and we'll go over that in class. All right, so Tuesday, don't worry about that. So I'll see you on Tuesday with these definitions, cohort 1, and on Thursday, cohort 2, I'm going to see you with these definitions, right? Okay, talk to you soon.